Good night students, I am Mayank Tiwari. Today we will solve test number 55 of the General Agriculture series. Today I was busy in the morning, therefore I thought this time I should make a video for you. So let's see question number 1. Culturable command area of minor irrigation project. You have to tell what is the cultural command area of minor irrigation project. Option A 2000 hectare, option B 2000 to 10,000 hectare, option C greater than 10,000 hectare, option D none. So student, this is very important. Please remember classification of irrigation project based on financial limit or expenditure. Option A, major irrigation project, which I gave you, if the project cost is more than 50 million rupees and if the cultivable command area is greater than 10,000 hectare, then it is called major irrigation project. More than 50 million rupees and cultural command area more than 10,000 hectare. This cultural command area also written as CCA. Please don't confuse it. And medium irrigation project is 2.5 million rupees to 50 million rupees and cultivable command area is 2000 to 10,000 hectare, and this minor irrigation project has a cultural command area 2000 hectare and the cost is less than 2.5 million rupees. So please remember these. Student, question numbers 2 you see, it is very easy question but can confuse you. Domestic sludge and sewage water is called as option A black water, option B grey water, option C fossil water, option D green water. So student, you have to tell what is this water known as, domestic sludge and sewage water. So option number D you can easily eliminate, student, green water, because green water is water found in plants. So please students remember blue water also. Blue water source is ocean, lakes, river, canal, and this type of water is exclusively used for irrigation. Green water, I told you, the water which is present in plant and the water which is present in soil moisture, it is green water. And fossil water is from water from kitchen and domestic waste. And student, green water you see, grey water is discharged from tanneries. And student, please also remember that these tanneries are also source of hexavalent chromium. Please remember that hexavalent chromium is produced by tanneries and electroplating industries. And student, black water, domestic sewage and sludge water, it is called black water. So please student remember this, and also remember that this type of water can be used for crop protection by giving some treatment to purify that. You cannot use black water directly for irrigation purposes. Now student, let's see some physiology questions. Physiology questions are often asked in various competitive exam. So you have to tell which is the first plant hormone to be discovered. Option A GA3, option B auxin, option C cytokinin, option D ethylene. So what is the first plant hormone to be discovered? So student, of course it's auxin. And also remember that cytokinin is also called kinetin, and ethylene is the only hormone which is gaseous in nature, and ethylene is also called fruit ripening hormone. Abscisic acid is ABA. So let's check quickly some facts about them. Student, first of all you have to remember bioassay of every hormone. Like bioassay of auxin is Avena curvature test, please remember it. And discoverer of auxin is Vent. And auxin is a Greek word which means to grow, please remember that. And auxin promote phototropism. This auxin also promotes geotropism. It also promotes cell elongation. Auxin cause cell division in vascular cambium. Auxin promote apical dominance. This is the most important property of auxin. And cytokinin counteract the phenomena of apical dominance. Please remember that. This tea plantation thing you might have read in class 11th. That in tea plantation decapitation is done so that auxin is removed and the lateral growth happens. And student, auxin prevent the formation of abscission layer. Auxin also controls xylem differentiation, student, please remember. Now some important fact about auxin. Foliar spray of NAA and 2,4-D cause flowering in lychee and pineapple. Please remember that. Lychee and pineapple, foliar spray of NAA and 2,4-D will cause flowering. Methyl ester of NAA cause preventing of sprouting of potatoes in storage. Please remember this. This is very commonly asked, that which is used for preventing the sprouting of potatoes in storage. Methyl ester of NAA, and 2,4-D is used as weedicide. It is also known as Agent Orange. Student, now some fact about gibberellin. Gibberellin was the second plant hormone to be discovered. It was discovered by Japanese scientist Kurosawa, and there were two Japanese scientists, Yabuta and Sumuki. They first isolated gibberellin from Gibberella fujikuroi fungus, and this Gibberella fujikuroi causes a disease called Bacane disease or foolish yielding disease of rice. Also remember the precursor of gibberellin, it is terpenes. And student, please remember gibberellin breaks seed dormancy. by mobilization of enzyme alpha amylase. Student, gibberellin also promotes synthesis of mRNA. Please remember that gibberellin promotes synthesis of mRNA and gibberellin promote etiolation. Etiolation means when the plants are kept in dark, they become pale yellow in color and they become abnormally long. This is due to gibberellin. It promotes etiolation. And gibberellin is a substitute of vernalization. Please student remember this. Vernalization is cold chili stimulus required for flowering. It is called vernalization, and vernalization term was given by Lysenko. Please remember that. And also student remember that foliar spray of gibberellin in genetically dwarf plant changes them to tall plant. And student, this is the most important property of gibberellin. Foliar spray of gibberellin promote induction of maleness in female plant. And student, this question is often asked that bolting in cabbage is caused by, it is caused by gibberellin. Please remember that. Now students, some fact about cytokinin also. Cytokinin was discovered by Miller and Skoog, and commonly cytokinin are BAP, benzyl aminopurine. I already told you that cytokinin counteract the phenomena of apical dominance. Cytokinin delay aging. This is called Richmond-Lang effect. Please student remember Richmond-Lang effect. Cytokinin delay senescence, and this effect is called Richmond-Lang effect. Coconut water is a resource of cytokinin. So if you keep anything in coconut water, it will remain fresh for a long time. This is called Richmond-Lang effect. Please remember this. And also remember that first cytokinin was extracted from herringfish sperm DNA. Please remember this, herringfish sperm DNA. First cytokinin was discovered from this. And student, please remember that first oxygen was discovered from urine of a human. The humans which suffer from deficiency of vitamin B3 or niacin, they suffer from pellagra. From their urine, oxygen was extracted. Please remember, oxygen was not extracted from a plant. It was extracted from a urine. And student, now some fact about ethylene. So its precursor of ethylene is your amino acid methionine, and ethylene causes fruit ripening. Ethylene promote leaf senescence. Ethylene increases respiratory rate during fruit ripening. It is called climacteric rise. Please student remember that. And ethylene promote horizontal growth of seedling, swelling of axis. Ethylene causes apical hook formation in dicot seedling. Ethylene promote root growth. Ethylene breaks seed dormancy. And ethylene promote formation of root hair. Ethylene promote sprouting of potato tuber. And ethylene inhibit flowering, except in pineapple. What is the bioassay of ethylene? Triple response to pea seedling is the bioassay of ethylene. And ethylene causes horizontal growth of seedling. This I already told you. Now student, this ABA, abscisic acid, precursor of abscisic acid is amino acid carotenoid. Sorry, student, amino acid carotenoid is not a amino acid. So please remember that. Precursor of ABA is just carotenoid. It is a plant pigment. And ABA has various names like inhibitor, abscissin, dormin. Abscisic acid is a growth inhibiting hormone. It promotes formation of abscission layer, senescence or aging, and mostly act antagonist to gibberellic acid. It promotes bud dormancy. Student, see question number 4. RQ for oxalic acid. RQ value, student, I already told you in previous lectures. RQ value is respiratory quotient. So it is asking you respiratory quotient value for oxalic acid. Option A are 1.33, option B 4, option C 0, option D infinity. You have to tell what is the RQ value of oxalic acid. So students, some fact about RQ value first. RQ is respiratory quotient, and volume of CO2 evolved upon volume of oxygen consumed. It is measured by Ganong respirometer. When organic acid are used as respiratory substrate, RQ value is more than 1. So please remember that. When oxalic acid is used as respiratory substrate, the RQ value is 4. Please remember that. And RQ value is 0 in case of incomplete oxidation of carbohydrate, as seen in case of succulent. For example, cactus, bryophyllum, opuntia. In their case, there is incomplete oxidation of carbohydrate. Therefore, the RQ value is 0. And student, in case of anaerobic digestion and fermentation, the RQ value is infinity because the volume of oxygen consumed is 0. Now student, last question of the day. Dicot leaf are option A hypostomatic, option B isobilateral, option C dorsiventral, option D A and C, option E all. So you have to tell what is the anatomy of dicot leaf, whether they are hypostomatic, isobilateral, dorsiventral. So student, please remember these terms. Dicot leaf are hypostomatic. So what is hypostomatic means? Number of stomata are more in lower epidermis as compared to upper epidermis to prevent transpiration. So please student remember hypostomatic. Dicot leaf are also dorsiventral. Monocot leaf are isobilateral and amphistomatic. It means number of stomata is almost equal on both the side of epidermis. And student also remember one test called Cobalt chloride paper test. It is used to compare transpiration on the surface of leaf, compare rate of transpiration, and also remember rate of photosynthesis is measured by IRGA, infrared gas analyzer. It is used for measuring rate of photosynthesis. And student also remember Ganong potometer. It is used for measuring the rate of transpiration, Ganong potometer. And one statement Curtis has given, transpiration is a necessary evil. So student, that's all for today. Thank you. Good night.